Hey guys, I just wanted to make a couple videos leading up to the exam of topics that people tend to have questions with or frequent topics that show up on past exams. So the first one I'm going to do is about osmosis, diffusion, and facilitated diffusion and water potential. So I'll try to make it kind of fast, but I want to make sure I cover it thoroughly because this is a topic that likes to show up on the exam. So under modules, if you go to the AP Bio review sheets, I've put a lot of these doodle diagrams that you guys can download to put in like a folder for quick reference on the exam. So I'm going to quickly go through this information with you guys. I do recommend reading over it prior to the exam and not just having it there in case you need it. That way you can remember what you actually have. But I'm going to go ahead and look at this one that talks about osmosis, and then we're going to look at the one about facilitated diffusion. So remember, diffusion of anything is just the movement of a molecule from an area of high concentration to low concentration. Um, so we talk about when we talk about perfume diffusing across a room or an air freshener diffusing across the room, it goes from an area where it's highly concentrated and it spreads out to an area where there's a low concentration. Osmosis is the specific movement of water from an area of high to low concentration, and it needs to be crossing through a semi-permeable membrane. So our cell membrane is a semi-permeable membrane. So typically we talk about water moving across the cell membrane in or out of the cell. So remember that leads us to three different scenarios. Hypertonic solution is one that has a high salt solution. So tonic, think salt, hyper, think high. So a hypertonic solution has high salt. Well, if there's high salt, there's low water, and water always goes where there's less water. So the reasoning for that is because if there's a lot of salt particles, all the water molecules are going to be busy helping dissolve it. So you can see here all these charged molecules are surrounded by water. So the water molecules are occupied and they're not free water molecules. And so we would say that the concentration of water that's available to do work is actually low because all of these are busy. So again, if these water molecules were like little workers and they're all busy dissolving something, and you go in and you say, hey, can I get some help? They're like, okay, well, we only have like 3 spare water molecules. So they just aren't, there's a low concentration of free water molecules. As opposed to if there was no solute dissolved in the water, then everything would just be kind of sitting around waiting to help. And they were like, hey, can I get some help? And they're like, sure, we're all available. So over here, there's no solute particles. So all of this water is available to do work, as opposed to over here, only a couple are available to do work. So we'd say water would move from an area of high concentration to low concentration. Another thing that I taught you guys is salt sucks. So all of these charged particles represent salt. And the term salt sucks means that salt is going to suck water in the direction of the salt. So wherever there's more salt, it's going to suck the water in that direction. So I think of the salt like at the end of a straw and it's sucking and the water goes in the direction of the salt. So wherever there's more salt, that's where your water is going to be. The reasoning being that high salt means low water. So the hypotonic solution, hypo, low, tonic salt. So this one is low salt, is going to be the one that is going to have a high water concentration. So hypertonic solution has a high salt, low water. Hypotonic has a high water, low salt. So isotonic means it has the same solution of salt in and out of the cell. So here we have 2 charged particles taking up some of our water molecules on each side. And so we have the same amount of free water molecules on either side. And there's no difference on either side. So water would not move from high to low. It would move back and forth across the membrane randomly, but there'd be no net change. Things that can affect the rate of osmosis being temperature, concentration differences, and how permeable is the membrane. Reverse osmosis, that's just like a little interesting fact. You can read over that. Remember that water is a polar molecule, so it has trouble moving across the membrane. Typically, only small nonpolar things can cross the membrane freely, but water is really tiny, and sometimes it can wiggle its way through. But because water has trouble crossing the membrane on its own, we have these embedded proteins in the membrane called aquaporins, which are going to allow water to move faster. This is an example of a facilitated diffusion. where this is a channel protein that allows water to pass freely and more quickly than it would be by wiggling through the membrane, because it's probably going to get rejected by the hydrophobic tails as it tries to wiggle through, and the rate of osmosis would be really low if there were no membrane proteins allowing water to cross. So this is going to greatly increase the rate of osmosis, or the rate of osmosis, and make water travel in the direction that it would from high to low much faster. So we have animal cells in a different solution. Remember, we are made of animal cells. Animal cells prefer to be in an isotonic solution so that the water going in and out of the cell is the same rate, and our cells do not swell or shrink, and they remain in equilibrium. If they're in a hypertonic solution, that means they're in a high salt solution. So the cell is submerged in a hypertonic solution. Hypertonic solution means the solution has high salt. So remember, salt sucks. It's going to suck the water out of the cell into the solution. So the water is going to leave the cell, go into the solution, causing the cell to shrink and shrivel. Animal cells in a hypotonic solution. That means they're in a low salt solution. So hypotonic implies that the solution of salt outside the cell is lower than the solution of salt inside the cell. So salt is going to suck. So there's a higher concentration of salt inside the cell. It's going to suck the water inside the cell, causing it to explode. So I have a little saying, hypo-hippo, meaning that in a hypotonic solution, a cell is going to swell like a hippo. So it could cause it to expand to the point of exploding, which will be an example of something we call cytolysis. Cytocell lysis, to break. So cytolysis is the breaking of a cell. Plant cells actually prefer to be in a hypotonic solution because when they swell, they push against their cell wall, causing them to be turgid, which is a name for just rigid, and allows them to stand up because they don't have bones. Plant cells in a hypertonic solution would also shrink, and they would pull away from their cell wall, which causes something called plasmolysis. They don't like that. That's really bad for plant cells, which is why we don't water our plants with salty water. You water plants with fresh water. And then here, plants in an isotonic solution, they just don't get that rigid structure, so it causes them to be kind of flaccid, and that's why your plants will wilt, is because they're not pushing against their cell wall to plump up, to actually stand up. So that's why you water plants with fresh water, is because the fresh water is going to be low salt. It's going to cause this situation to happen where they will actually cause your plants to stand up as opposed to wilt. And then up here, we would not put fresh water directly into our veins, because if we put fresh water directly in an IV into our veins, we would have this situation occurring, which is no good, and our cells would explode and we would die. As opposed to when we put an IV, which is isotonic with our body, it's going to cause our cells to remain in equilibrium. Okay, so now I want to talk about facilitated diffusion. So if we scroll down, it's the note sheet before this. Okay, so facilitated diffusion is just, again, it's the movement of a molecule from high to low along its concentration gradient, but it's with the help of a membrane or a channel protein that allows the molecule crossing the membrane to cross more freely. Remember, the membrane is made of phospholipids arranged in a bilayer with hydrophilic heads and hydrophobic tails. So the majority of the membrane is hydrophobic nonpolar. This whole section is nonpolar hydrophobic. So the only things that can freely pass through are small nonpolar things. So when something that's large, charged, or polar wants to cross through, we have these help of a membrane protein that allows them to go in the direction of their concentration gradient, but it's like a door. So think of the membrane as like the walls of a building. So we'll think of like Providence, and around the building we have doors, points of access that things can go freely in and out along their concentration gradient. And so you can't just walk through the brick walls because you're large. So you have to go find a point of access, which would represent a membrane protein. So it's diffusion because you're going in the direction that you want to go, but it is facilitated, meaning it's helped out by a protein that allows it to pass through. So an example of facilitated diffusion down here is talking about how some channels are gated and that they can open and close based on the conditions around the membrane. So an example of gated ion channels are ones in your neurons that need to have something bind to the outside of the cell to open the protein so that the things can pass in and out. So some aren't just open all the time. They're not just open to the public. They need to have something that causes them to open specifically for certain molecules and during certain conditions. So I use the example up here of Providence having doors around the building. Well, we don't necessarily want the doors to be available to everybody all the time. So something that could bind to the exterior of a protein and cause it to open and close will be like our security badge. So here, in the presence of a ligand, it can cause the channel to open or close. And so the ligand would represent like the security badge that binds to the outside of the membrane protein or in this receptor right here. And when the ligand binds to the receptor, it can cause the door to be open or closed. So in this case, with the ligand bound, it's actually causing the protein channel to close. So ligands are molecules that can stick to the channel and open or close it like a key in a lock. So again, the ligand could represent like the security badge regulating who can go in and out at what times. So your security badges are deactivated at different times of the day. And so you can open and close it at different times. And that's just kind of the function of a ligand, is to regulate when these molecules can go in and out. So why does that apply to cells? During when your neurons are firing to cause your brain and nervous system to work, a neurotransmitter is going to bind to the outside of the cell. And when that neurotransmitter binds, it acts as a ligand and it opens these sodium channels. And when the sodium channels are open, it allows an action potential to fire, and it actually causes your cells to work together and kind of like electricity to travel through your cells for your brain and spinal cord to work. And so neurotransmitters are little messages that arrive at your brain cells, and when the ligand or the neurotransmitter binds to the outside of the cell, it causes the sodium channels to open, and when the sodium channels open, an action potential can fire. And so this is kind of how cells regulate. When something comes and binds to a receptor, it can cause an ion channel to open, and then when the ion channel opens, things will go from an area of high concentration to low concentration. And if we're looking at ions, sometimes that causes a change in the charge of the cell on the outside and the inside. And when you change the charge, it can cause kind of like a chain reaction to occur. So this would be an example of like cell communication, where this ligand binds, causes the ions to go through the cell. And when the ions go through the cell, they're going to change the charge of the inside of the cell, which can cause something like an action potential to fire, or maybe it could cause a cascade effect that results in gene expression. And so that could be another example of just like a cascade effect because something binds to the outside of a membrane protein. Okay, this one's interesting. It's a kind of an example of what could happen if these are not working. So I could definitely see this being in like an FRQ about what would happen if your membrane proteins weren't working correctly. And so here we have an example of a functioning chloride channel in the lungs. So in people with functioning chloride channels, the chloride channels are open so that the chloride ions can flow out. So typically the chloride channels are open and the chloride is going from high to low, high concentration inside the cell to low concentration outside of the cell. So it's moving out. Remember that chloride would be an example of a solute or a salt. So the direction that the solute is moving is the same direction that water is going to move. So if salt's going to suck the water along with it, so the salt's going to move out, the water's going to move out with it. So as chloride ions flow out, water molecules also flow out, allowing the mucus that builds up outside the lung cells to stay wet and thin. So outside the cells of the lung, so this represents the outside of the cell in the lung, as this chloride comes out, water's going to come out, and any mucus that's here is going to get thinned out by the water so that you can just like cough it up. people who have cystic fibrosis have a mutated or non-functioning chloride channel. So this is a membrane protein that has the wrong shape. So when you think about this, think, okay, there was probably a mutation in the DNA. That mutation in the DNA was transcribed and translated, and the protein did not fold correctly. And when the protein was inserted in the membrane, because it was folded improperly, it's not going to, it's now a different shape. Its function isn't going to be the same, and it's not going to function properly. So the mutated protein here is preventing chloride from exiting like it normally would. And so when chloride can't leave, it's going to build up inside the cell. And when it builds up inside the cell, salt's still going to suck water in the same direction. So now instead of water leaving with the chloride, water's going to go in the direction of the chloride, causing the water to come in the cell. Individuals with cystic fibrosis, their chloride channels don't allow ions to flow out. So since the ions build in, water also comes in. And then the mucus outside is not able to be thin, so it gets thick and sticky, and it can build up and make them very sick. So because they cannot thin out this mucus, because the water is actually leaving the mucus and going inside the cell, and it's not getting thinned out by water exiting the cell, then it's going to get really yucky and hard to cough up. So it leads to this thick and sticky mucus, frequent lung infections, and that would be an example of facilitated diffusion in the cells. And then what would happen if the protein that would allow the chloride to pass through was mutated. So again, when I see this, my brain automatically goes to transcription and translation and DNA, thinking, okay, well, the DNA was mutated, so the protein is mutated. Now that the protein's mutated, it has the wrong shape. And what's going to happen, see, the difference between these two shapes tells me that this different shape is going to cause different functions in these membrane proteins, and the different function in this case is a bad function because it's not allowing people to actually process the mucus and stuff and cough it up and all that good stuff. So that is diffusion, osmosis, and facilitated diffusion. I'm going to make another video about water potential in just a second, so make sure you follow up this video with the water potential video.